Hey everyone, Azim here. We are taking a look at a model of a cardiac contractile cell, a muscle fiber that you find in the heart. Now cardiac contractile cells, they're not skeletal muscle. Skeletal muscle would be long, very, very long and multinucleate. They're not like smooth muscle. Smooth muscle are shorter and more spindle-shaped. Cardiac contractile cells are just like skeletal muscle, though, in that they have striations. You have these regular patterns of the banding of the proteins within sarcomeres. So that is something similar that they have. But cardiac contractile cells, there is one other cardiac myocardium type. It's called autorhythmic or pacemaker cell. That's different. That's not for contraction. That's for setting the pace. These are for contraction, moving blood around in your heart and having it go from your atria to ventricles and from your ventricles to either pulmonary or systemic circulation. So shown here is one, at least on here on one side, we have one individual cell. This is all one cell. And then over here, we've got a fragment of another cell. So looking first at this one cell, you can see that it's branching. You can see a branch here. The sarcolemma comes around and wraps around and then goes here and then comes down and would connect with another cell over here. So it has a sarcolemma just like any other muscle fiber. It has T-tubules, so you can see the holes from the surface. You can see the purple stuff here. Those are representing T-tubules. It has sarcoplasmic reticula that release calcium ion into the sarcoplasm. So shown here in the darker blue, that is the sarcoplasmic reticulum. From here to here, that is one sarcomere. So you've got the Z line, you've got the thick filaments. Sorry, you've got the M line, you've got the thick filaments here in this lighter green. You've got Z lines on either end, and then thin filaments are matching in between that. So these sarcomeres repeat over and over and over again. T-tubules and sarcoplasmic reticula kind of wrap or straddle around it. Lots of mitochondria. You need lots of mitochondria and myoglobin to supply oxygen to these cells that are working all the time. But one other thing that distinguishes, so all those things that I just mentioned were very similar to skeletal muscle, but what makes it different? One, that it's branching, but two, at those branching points, this region, it's called an intercalated disc. At intercalated discs, we can find some special features. One thing we can find is a desmosome. A desmosome is where the plasma membranes interlink. So you've got special proteins that link the two cells together. It's like a hitch. So if one contracts, if one shortens, then the other one's going to be pulled with it. We want to ensure rhythmicity in our heart, and one way to ensure rhythmicity is to link them physically with a desmosome. Another thing that can link them, not physically but electrically, is a gap junction, which I don't think is shown here. Yeah, not really shown here. But we have desmosomes and we have gap junctions. Gap junctions are channels between two cells so that ions can flow, like sodium ion can flow from one cell to the next. So if we have a muscle action potential in one cell, that muscle action potential will carry over to the next cell. So we're ensuring rhythmicity, one cell to the next, to the next, to the next. This is a longitudinal cut. Oops, sorry. Longitudinal cut here is a cross section. You can see the myofibrils within the muscle fiber all bundled together to form a cardiac contractile cell. All right, that's that. Please let me know if you have any questions.